So we have three things. I'm going to give you an example of antibiotic-resistant microorganism, which is going to be Mycobacterium tuberculosis eventually, probably not now. What we're going to do, we're going to talk about how you test for antibiotic efficiency, and maybe mention some of the resistance, maybe not. Actually, you know what? Now let's talk about resistance. It's going to be better because I can talk about resistance meaningfully, and then maybe segue into. So what are the main? First of all, antibiotic resistance. Being antibiotic resistance is a phenotype. Does everybody agree with that? It's a phenotype, which means it is determined by the presence of certain genes. Are you following me? So it is in the code. Now, the sources of antibiotic resistance in bacteria, it's not a great classification, but it kind of gives you an idea. So one, sometimes it's called intrinsic resistance, and another type of resistance is called acquired, although, quite frankly, both of them are acquired. It depends when and where. So let me explain the difference. What is an intrinsic antibiotic resistance? So imagine you have a patient who was, I don't know, hiking in the mountains, fell, had a wound, and now wound is infected by some environmental microorganism from the soil. Are you following me? This microorganism was sitting in the soil for years and years, right? And it was fighting and competing with other organisms. Does that make sense? And it developed its own set of tools to compete, to kill other bugs. Does that make sense? So this microorganism not only kills other bugs by its antibiotics, it also potentially has a resistance to antibiotics produced by competitors. Are you following me? So you can isolate a microorganism from the soil and find out that suddenly it is resistant to fluoroquinolones. For some reason. It was never exposed to them, it's just resistant. This is intrinsic. Are you following me here? Like, it happened, it's just in the genome of that microorganism. There was, humans were not involved in that particular trait. What is acquired? Again, acquired is, So imagine that you are, what is the issue with antibiotic treatments? When you use antibiotics to treat infections, you want to make sure that you nuke each and every cell. Am I clear? Because what you do, you apply a selective pressure. Does that make sense to you? Once you apply selective pressure on the bacteria, the ones that are more resistant, the ones that survive, will be selected. Why they survive? I don't know, they have some kind of mutation. Something happens. They are more resistant to antibiotic. Does that make sense? This is what I call acquired. We have Enterobacteriaceae that are resistant to carbapenems. Why? Because we used carbapenems maybe in suboptimal doses, not for a long time, this and this and that. Am I clear? Does that make sense? No? Good?
So, like an example would be like, so children get ear infections a lot. So then they push amoxicillin for ear infection. And then they're done with the 10-day amoxicillin, and then a week later, ear infection comes back. So then they do a different type of antibiotic, and then no more ear infection.
Say the last statement.
And then the ear infection finally goes away.
So what, in the example you described, when they use amoxicillin, the infection comes back, maybe the dosage was not optimal, and They killed most of the microorganism, right? But some of it is left, and what is left is resistant. Does that make sense? Like survival of the fittest. You're absolutely right. And will amoxicillin work on those fittest? A larger dose, longer treatment, possibly. But again, you have to consider what's going to kill your patient first, right? Your antibiotic or infection. You see what I'm trying to say? So, one thing that I want you to be clear about, when we say resistant, it's not Hulk or Superman resistant. It's not impenetrable. Okay? It means conventional dose doesn't work. And you can increase the dose only so much. Am I clear? So, let's talk about the mechanisms. I'm going to be very, very clear about the mechanisms of antibiotic resistance. There's 7 of them. Well, 8 that I want you to know. I'm going to list them and explain what they are. So, drug modification. Great example, beta-lactamases. They chemically modify beta-lactams, rendering them inactive. Does that make sense? So this is when bacteria modify the drug. Now, drugs, antibiotics, they all have targets. Are you following me? So, target modification is another strategy. Good? For instance, have you heard about MRSA, methicillin-resistant Staph aureus? So methicillin inhibits an enzyme in the Staph aureus. Follow me. In MRSA, this enzyme is different, and methicillin can't bind to the active cycle, or binds very weakly. You see what I'm saying? So, target of methicillin is modified, rendering methicillin useless. Good? Inhibition of uptake. You basically close the door for the drug. Efflux pumps. You pump the drug out of the cell. Enzymatic bypass. So let's say, remember we were talking about those metabolic pathway inhibitors? Sulfamethoxazole, trimethoprim. So now bacteria just take a detour enzymatically. So this route is inhibited, but there is some other pathway that can still be used. Does that make sense? Target overproduction. Again, I will have to jump back to trimethoprim, sulfamethoxazoles. Remember folic acid synthesis inhibitors? So those inhibitors are competitive. Are you with me? So they competitively inhibit those 2 steps. If you simply produce a whole bunch of enzymes, like a lot of enzymes that those 2 inhibit, they can inhibit only 1%. You see what I'm trying to say? 99%, you just can't give patient enough drug to inhibit every molecule. One of my favorite ones, it's called target mimicry. So this is a really fancy way. Bacterial cell, produces a decoy molecule that would bind the drug instead of a target. You see what I'm saying? So this is one of the mechanisms for fluoroquinolones resistance. Bacteria would produce, I think it's some DNA molecules. So basically some decoys. So ciprofloxacin would bind to decoy and would not bind to the target, and target will still be workable. Does that make sense? Last mechanism is mini cells. I just find it absolutely crazy fascinating. So in case of mini cells, this has been shown recently. It's really cool. So remember we were talking about aminoglycosides, how they result in the production of faulty proteins. So you've got a whole bunch of crappy proteins in a cytoplasm of the cell. Does that make sense? So what cell does, it packages those faulty proteins together with antibiotic into small vesicles. It was recently discovered that bacteria can do that. Like packaged in a tiny little trash bag. And they just release this trash bag. Okay, we're good? So, what do you need to know about these guys? I give you a description. Okay? Like I tell you, in methicillin-resistant Staphylococcus aureus, transpeptidase is modified so that beta-lactams cannot bind to it. What kind of resistance mechanism is it? Tell me, okay, if a target, so what is transpeptidase? A target for methicillin. It is modified, it's a target modification. Does that make sense? This kind of stuff. Last bit, which is a conceptual bit, an important conceptual bit. So, all of these mechanisms require an effort. Are you following me? Do you understand what I'm trying to say? So take two strains of bacteria, one that's resistant, let's say it can do drug modification, and one that is not. Listen, the one that does drug modification has to produce an extra enzyme, which requires resources, energy, amino acids. Does that make sense? Space in the genome. So it's kind of an interesting thing, but without antibiotic in the environment, being antibiotic resistant is a disadvantage because it takes the resources off. Do you understand what I'm saying? Do you understand? And if you will grow a resistant and non-resistant strain side by side, like in the same vial, without antibiotic, resistant and non-resistant, in the same vessel without antibiotic, which one will grow faster? What do you think? Without antibiotic, resistant versus non-resistant, which will grow faster, which will be more successful? Non-resistant, because it doesn't have to waste resources on something that is not necessary at this particular moment. Does it make sense? Questions? Okay, so we're going to finish that stuff next week.